Good morning, all. Welcome back to our official YouTube channel, SR and BGNR Government Arts and Science College, Khammam. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. We are discussing about the fourth unit in third semester of biochemistry course, that is regulation of enzymes and their inhibition mechanisms. In the last class, we have been discussing about inhibition of enzymes. The enzyme inhibition is broadly divided into three different categories: reversible inhibition, irreversible inhibition, and allosteric inhibition. In the last class, in the previous video lesson, we have discussed about the reversible inhibition. Let us quickly go through the previous class and recap it. First of all, inhibition is means inactivating the enzyme, inactivating the catalytic activity of the enzyme. If a molecule binds to enzyme and inhibits its function, that molecule is called an inhibitor. The inhibitor may bind to active site or other than the active site also. So based on that, different mechanisms have been established. So in those different categories of inhibitions, in the last class we have discussed about the reversible inhibition. Reversible inhibition means the inhibitor binds reversibly to the enzyme. That means it may bind to enzyme and at the same time it may dissociate also easily. So here the inhibitor cannot bind tightly by covalent bonds. It binds the enzyme by means of non-covalent interactions which are very weak. Like substrate interacts with the active site of the enzyme, inhibitor also interacts with the enzyme with non-covalent interaction. So that is why it is called reversible inhibition. So we can get the active enzyme from inhibited state. So if the substrate concentration is more, the enzyme will be active. If the inhibitor concentration is more, the enzyme will be inhibited. In that way, we can get active enzyme from the inhibited form also. That is why we call this kind of inhibition is a reversible inhibition. In reversible inhibition, there are two subcategories based on the site of the inhibitor bindings, based on inhibitor binding site. First one is competitive inhibition and second one is non-competitive inhibition. In competitive inhibition, the inhibitor binds to the active site like substrate. It binds to the active site, so it competes with the substrate for the active site. Therefore, it is known as competitive inhibition. The inhibitor will have similarity in structure with the substrate, therefore it is binding to the substrate binding site. So here we have to consider a few important points when we are talking about competitive inhibition. First one is the substrate is having similarity in structure with the substrate inhibitor, therefore inhibitor can also bind to the active site by non-covalent interactions. If the concentration of inhibitor is more, then inhibitor will have a greater chance to bind to active site. If the concentration of substrate is more, then the substrate may bind to the active site more than the inhibitor does. So therefore, it depends on the relative concentration of the substrate and inhibitor. In this case, as the inhibitor is binding to active site, competing with the substrate for the active site, the Km value increases. But highest concentration of substrate levels, the Vmax, the enzyme approaches Vmax. So therefore, there is no change in Vmax. In competitive inhibition, Km value increases and Vmax remains unchanged. One best example for competitive inhibition is succinate dehydrogenase enzyme, which convert succinate to fumarate. This enzyme can also bind malate, that is malonic acid, malonates, glutaric acid, and oxalic acid. Similar kind of dicarboxylic acids can also bound by SDH, succinate dehydrogenase enzyme. So these three different dicarboxylic acids act as competitive inhibitors for SDH. And second one is non-competitive inhibition. In non-competitive inhibition, the inhibitor is completely different from the substrate. It will not compete with the substrate for the active site. It binds other site other than the active site on the enzyme molecule. So it will have separate site for binding. Therefore, it binds to other site and inhibits the enzyme activity. So it can bind either a free enzyme or enzyme substrate complex also. It is also by means of it is also binds by means of non-covalent interactions. There is no similarity between substrate and the inhibitor. And so one of the best example is mercury ions, which binds to the to sulfhydryl containing enzymes. If the enzyme is having a sulfhydryl group other than the active site, then the mercury ions binds to those sulfhydryl groups, and this binding will induce a conformational change in the active site of the enzyme. Therefore, the enzyme will be inhibited. In this case, the inhibitor is not competing with the substrate for the active site. Therefore, the active site strictly binds substrate only. That means the Km value is unchanged. But the velocity of the enzyme will be affected due to the binding of inhibitor. Therefore, the Vmax is always lowered in the presence of inhibitor. And the second kind of inhibition is irreversible inhibition. So far we have discussed about reversible inhibition, where we can get active enzyme from the inactive enzyme, inhibited enzyme, by removing the inhibitor from the active site or uh, other sites where it binds. But this kind of enzyme inhibition is irreversible inhibition, where we cannot get active form of the enzyme from a inhibited form because the inhibitor binds by means of covalent interactions, not by the non-covalent interaction. Therefore, once the inhibitor binds to the enzyme and inactivates it, then it will be permanently inactivated. We cannot easily remove the inhibitor from the enzyme because it bound by means of covalent interaction. So that is why it is called irreversible inhibition. So here the inhibitor binds covalently This is very important point, that the inhibitor binds covalently with the enzyme and inactivates them, which is irreversible. The inhibitors are usually toxic, poisonous substances. So these irreversible inhibitors are known as toxic poisonous substances because they are permanently impairing the activity of the enzyme, and those enzymes may be essential for the life. So if they are very important for the maintenance of life, then those important enzymes will be inactivated. So that will be that will cause a potential danger to the organism. That is why these kind of irreversible inhibitors, also known as toxic and poisonous substances. For example, iodoestate. Iodoestate is an irreversible inhibitor of the enzymes like papain and glyceraldehyde 3-phosphate dehydrogenase. Here, a papain is a hydrolytic enzyme, glyceraldehyde 3-phosphate dehydrogenase. This is one of the key enzyme in glycolysis. These two enzymes are having sulfhydryl group in their active site. Okay, they have sulfhydryl group in the active site because they are having a cysteine residue in that active site. So this is very much important for the catalytic activity. So here the irreversible inhibitor iodoestate simply comes and binds to sulfhydryl group by means of a non a covalent interaction. So therefore the iodoestate will be in the active site, that doesn't allow binding of their own substrates. Therefore these enzymes will be permanently impaired. And another important example for irreversible inhibition is DFP, diisopropyl fluorophosphate. This is also called as nerve gas, which is developed by Germans during Second World War. They have developed a gas-like substance which is known as diisopropyl fluorophosphate, which is used as a irreversible inhibitor. So the enzymes which are affected by this diisopropyl fluorophosphate are serine proteases and acetylcholine esterases. Cysteine proteases like papain are affected by iodoestate, and serine proteases are affected by diisopropyl fluorophosphate. So in this case, this diisopropyl fluorophosphate binds to the serine hydroxy group of the serine in the active site of the enzyme, and at the same way it also inhibit irreversibly the acetylcholine esterase, which is very important for the conduction of nerve impulse. And one more important example that we generally observe is many organic phosphorus insecticides. Whatever pesticides or insecticides we are using in agriculture field, or most of them are irreversible inhibitors. They will block the activity of acetylcholine esterase, which is essential for nerve conduction. So in the presence of these irreversible inhibitors like the organic organophosphorus insecticides, they will inhibit the acetylcholine esterase activity, and they thereby impairing the nerve conduction. The nerve impulse transmission will be disturbed, and due to failure of nerve nervous system, the insecticide dies. So that is the mechanism of irreversible inhibition by organophosphorus insecticides. Apart from using these irreversible inhibitors as poisonous and toxic substances, in research also we can use them for the identification of the residues which are present in the active site of a given enzyme. Suppose if we have an enzyme and we want to characterize it, first of all, we wanted to find out what are the residues responsible for the catalytic activity of the enzyme. We want to know. For this, we can use these kind of irreversible inhibitors which binds the active residues present in the active site by covalent interaction. So once we have bound inhibitor to the active residue in the active site, we can hydrolyze the entire enzyme molecule into constant amino acids. Then we will get a amino acid which is linked by the irreversible inhibitor. So that amino acid is the key amino acid residue in that enzyme active site. So that way we can find out what are the key amino acid residues present in the active site. And very important thing is suicide inhibition. This is also one kind of irreversible inhibition, but the important point in suicide inhibition is these are specialized form of irreversible inhibitors. In this case, the original inhibitor binds to the active site of the enzyme and undergoes a few catalytic steps which convert it to a more potent form of inhibitor. So that means earlier we have seen uh, three different examples like iodoestate, diisopropyl fluorophosphate, and organophosphorus pesticides. All these irreversible inhibitors binds to the active residue present located in active site of the enzyme. Just they bind by means of covalent interaction. But here the inhibitor, in case of suicide inhibitor binds to the active residue in the active site by means of covalent interaction, and at the same time the inhibitor few steps undergoes a few steps in normal way. Therefore, the inhibitor is converted to another molecule and which acts as a potentially potential inhibitor to the enzyme. So whatever molecule we are using as inhibitor, it is not directly acting as inhibitor, but it is converted into other form which is very potent molecule as inhibitor. So therefore, the enzyme undergoes first few steps normally. Then the enzyme activity will be inhibited by the generated potential inhibitor molecule. So this is called suicide inhibition. Here the enzyme reaction is terminated prematurely. A very good example of suicide inhibition is allopurinol. Allopurinol is a medicine prescribed for the treatment of gout. So uh, in a gout patient, what happens? Accumulation of uric acid crystals takes place in the joints of the patient. The joints of the patient, the synovial fluids present in the joints will be filled with crystals of uric acid. Uric acid is nothing but the metabolic end product of purines. So if the concentration of uric acid is more, they get deposited in the joints, which gives symptoms of gout. They will experience uh, tremendous pain in their joints. So to relax them and to treat them from this symptoms from the symptoms of gout, they will be prescribed with allopurinol. Allopurinol is a suicide inhibitor which binds to its enzyme known as xanthine oxidase. That xanthine oxidase binds allopurinol as a suicide inhibitor, and it converts the allopurinol to alloxanthine. So alloxanthine is the dead end for the xanthine oxidase. Xanthine oxidase no more further carry out the reaction process to form the uric acid. It cannot form uric acid from the allopurinol. Allopurinol will be converted to alloxanthine only, not the uric acid, because it is a inhibitor. It is not the actual substrate for the xanthine oxidase. So because of that, allopurinol, alloxanthine, which is formed inside the active site of the xanthine oxidase, that tightly binds the xanthine oxidase, thereby inhibits the enzyme. So because of inhibition of the enzyme, uric acid production will be decreased, which eases the symptoms of gout. So far we have discussed about the inhibition of enzymes. Mm, there are three different inhibitions: competitive, reversible, irreversible, and allosteric inhibition. So far we have discussed about reversible and irreversible, and now we are going to discuss about the allosteric inhibition mechanism. Allosteric inhibition. When we look at the term allosteric, it means allo means other, steric means a region, a place on the enzyme. So that means the inhibitor, the allosteric inhibitor, is a substance which binds to a site other than active site. So if it binds to the active site, it may be a substrate, or if it is an inhibitor, it may be a competitive inhibitor. But in allosteric inhibition, the inhibitor binds other than the active site. So therefore it is called allosteric inhibition. It looks like the allosteric inhibition and the non-competitive inhibition both are same, but there are certain differences between them. In allosteric inhibition, the enzymes are very complex, and most of the allosteric the enzymes which are regulated by allosteric mechanisms, are oligomeric in nature. So they use those. That means the enzymes will have more than one subunits. Some subunits for catalytic activities, and remaining subunits for regulatory activity. So we will have regulatory subunits and catalytic subunits separately in allosteric enzymes. But in the case of non-competitive inhibition, we may have a single subunit or single polypeptide chain to which subunit binds at the active site, and the inhibitor binds other than the active site on the same polypeptide chain. But here it is very well organized. The subunits are organized into regulatory subunits and catalytic subunits. The allosteric effectors, the inhibitors, actually they are called as allosteric effectors. We have activators and as well as inhibitors. Allosteric molecules, the molecules which activate the enzyme by binding to the site other than the active site, they are called allosteric effectors or activators. They increase the enzyme activity by binding to them. And negative effectors, they bind the enzymes and decrease their catalytic velocity. They are called negative negative allosteric effectors or allosteric inhibitors. So we can have both positive regulation and negative regulation. If the enzyme binds activator, allosteric activator, then the enzyme activity will be enhanced, or otherwise if it binds inhibitor, then the enzyme activity will be decreased. So this is very much important for the regulation of metabolic pathways. As already said, most of the allosteric enzymes are oligomeric in nature. So we have subunits, more than one subunits. We will have more than one subunits in allosteric enzymes. Some are dedicated for regulatory activity, where the activators and inhibitors binds, and some are dedicated for the catalytic activity, where the substrate binds. The subunits may be identical or different. The non-covalent reversible binding of effector molecules at allosteric site brings about a conformational change in the active site of the enzyme, leading to the inhibition or activation of the catalytic activity. So that means, suppose we have an enzyme, we will have a subunit, sorry, substrate binding site here, and allosteric modulator may bind here. Suppose once the activator binds this allosteric site, it this brings a conformational change in the active site, which induces the binding of substrate to the active site. So therefore, the activity of the enzyme will be increased. So in the presence of activator, the enzyme activity will be increased. In the presence of inhibitor, allosteric inhibitor, if we have allosteric inhibitor, this inhibitor binds to the same regulatory site where the activator earlier bound. The same site where activator binds, the inhibitor also binds. If the concentration of inhibitor is more, then it binds to the regulatory site and it brings a conformational change in the active site, so that the active the active site can no longer binds the substrate. Therefore, the activity of the enzyme will be decreased. In this way, the activity or the rate of catalytic velocity of the enzyme depends on the allosteric modulator. If it binds activator, the catalytic velocity will be increased. If it binds inhibitor, then the catalytic velocity will be decreased. So, in the mechanism of allosteric inhibition, we have a model known as concerted model, where it explained that the allosteric enzyme exists in two different conformation. The entire enzyme exists in two forms. One first form is T form and second one is R form. T stands for tense or taut and R stands for relaxed. Tense means that is not at all flexible. It is completely at tense. So this is a an inactive form of the enzyme, and R is relaxed, where the conformational changes easily takes place and the active site easily binds the substrate. So that means R is the reactive form of the enzyme and T is the inhibited form of the enzyme. So therefore, all enzymes, allosteric enzymes, exist in either T form or R form, depending on the presence of activator or inhibitor. So here the R and T states are in equilibrium. Allosteric inhibitors favors T state, which is inactivated form, whereas activator and substrate favors or state. The substrate can bind only R form of the enzyme because it is relaxed, and substrate binding site, the active site, is accessible for the substrate. The substrate easily binds to the active site. Therefore, the R form is the reactive form to which the substrate binds and catalytic activity takes place. So, R straight is the reactive form, and it is the active form of the enzyme. So, substrate only binds to the R form, and the concentration of the enzyme molecule in the R state increases as more substrate is added. Therefore, the binding of the substrate to the allosteric enzyme is said to be cooperative. So, here cooperative means if we add some amount of substrate, that substrate will bind to the active site and increases the velocity of the catalytic activity of enzyme. If we add further, if we add the substrate furthermore, then the increased amount of the substrate will increase the R form. From T form to R form formation takes place. So gradually the equilibrium will be shifted from T state to R state. So therefore more number of enzymes will be in R form and they carry out the catalytic reaction. So that way the substrate is not only involving in the catalytic reaction, but it is also involving in the transformation of T state to R state. There, in that way, it is a cooperative uh, cooperative mechanism of activation of enzyme. When we look at the enzyme kinetics of allosteric enzymes, we will have a sigmoidal curve instead of a hyperbolic curve, which we get for non-allosteric enzymes. Here we can easily observe that here the enzyme is there. This is the enzyme, and we have active site here and allosteric site is here. Allosteric site is different from the active site. So if the inhibitor, this one is the inhibitor, if inhibitor binds to the allosteric site, it brings a conformational change in the active site. So therefore the substrate, the, the active site will be altered. The substrate is unable to bind. It is unable to bind to the active site of the enzyme. This way the enzyme activity will be inhibited. And instead of inhibitor, if we have activator, if the concentration of activator is more than the inhibitor, then activator binds to the allosteric site. The same allosteric site binds the activator. This brings a conformational change in the active site of the enzyme, which now easily binds the substrate and it carry out a catalytic reaction. So here, because of the inhibitor, because of presence of the inhibitor, this enzyme which is already in R form, it is transformed to T form, and now the T form which is transformed to R form in the presence of activator. So the R form can bind the substrate, whereas the T form cannot bind. And when we look at the kinetic behavior of allosteric enzymes, we will get a sigmoidal, this red color curve shows the sigmoidal curve, which is obtained by the kinetics of allosteric enzymes. Here, a cooperative activity takes place. Here, at the beginning of this reaction, the substrate levels are very low. As we add more amount of substrate, that substrate will gradually convert the T state to R state. So we are getting more amount of R state, R form enzymes. So as we add more and more amount of substrate, we will get more amount of R form enzyme molecule, and they will catalyze the reaction and produce the products. So it will, here the R form is less. Here you can see at low substrate levels the R form is less. As the substrate concentration increases, the R form is more, therefore more product is formed. And further increase in the substrate levels, what happens? The enzyme will reach the Vmax. Therefore, further increase in the substrate concentration will not increase the velocity of the enzyme anymore, because all the uh, substrate binding sites, active sites, are saturated with substrate. So that is why we will get a sigmoidal curve instead of a hyperbolic curve. So here a cooperative effect is taking place, that the substrate not only involved in the formation of product, but it also acting as a positive effector which converts T state to R state. Feedback regulation is one of the best example, one of the best process to explain the mechanism of allosteric inhibition. Feedback inhibition means the process of inhibiting the first step of any metabolic process by the final end product in a series of enzyme catalyzed reactions of metabolic pathway is referred as feedback mechanism. So we have an example here. Here we can see that, suppose this is a metabolic pathway. Suppose say for example it is for synthesis of an amino acid. E is an amino acid, and this metabolic pathway is for the production of E from precursor A. So A is the precursor. So here the A first is converted to B, B to C, C to D, D to then E. So in a series of reactions of metabolic pathway, A is ultimately converted to E. Each and every step involves separate enzymes E1, E2, E3 and E4. Suppose we need 100 units of a particular amino acid 1. If the cell is having only 50 molecules, so that means it is having, it is short of 50 molecules short of the required amount. So therefore it must synthesize the 50 molecules of amino acids. So in this case, what happens? The precursor is converted to B, then C, D, E. So the entire metabolic pathway goes smoothly as the amino acid synthesis is necessary. Once the amino acid concentration reaches the required saturation level, then the metabolic pathway must be stopped. Otherwise, it will it will continuously synthesize and waste the cellular energy for the production of this amino acid. So beyond 100 units, it is not at all necessary, and it will be a wasteful process when we consider the cellular economy. So therefore it must be stopped once the concentration of amino acid reaches 100, the required concentration. So in this way, in this case, what happens that E acts as an inhibitor for the enzyme E1. Here E1 is the key enzyme which involved in the regulation of this metabolic pathways. So for almost all metabolic pathways, the first enzyme will be the key regulatory enzyme, which can be which will be regulated by the final end product. Okay, here the E acts as an allosteric inhibitor and binds to E1. Therefore, E1 becomes inactive. Once E1 is inactive, A is not converted to B, and therefore the entire the complete pathway will be shut down. So this way it acts as a feedback regulatory mechanism. The final end product is inactivating the first step. This is called feedback regulation. So this mechanism comes under the allosteric regulation. ATCase is well studied best example for allosteric regulation process. ATCase means aspartate transcarbamylase. So this is the enzyme which is the key regulatory enzyme in pyrimidine biosynthesis. Here the reaction of ATCase is the enzyme will have an active site where it binds carbamyl phosphate substrate 1 and then aspartate substrate 2. It binds these two substrates and combine them to form N-carbamyl phosphate, N-carbamyl aspartate. This is the product of this ATCase enzyme. So here the enzyme is the first enzyme involved in the pyrimidine biosynthesis. So therefore the amount of pyrimidine which is the end product of pyrimidine biosynthesis, that is CTP. CTP is the end product of pyrimidine biosynthesis. So therefore the enzyme ATCase must be regulated by CTP by feedback mechanism. Otherwise it will continuously synthesize the CTP beyond the necessary levels. So that becomes a wasteful process uh, for cellular economy. So therefore ATCase is allosterically regulated by activators and as well as inhibitors. So in this case ATCase is an allosteric enzyme which have both T form and R form, tense form and that relaxed form, which is active form that binds to the substrate. So here we have two substrates, substrate 1 and substrate 2. Substrate 1 is carbamyl phosphate and substrate 2 is aspartate. During ATCase reaction, carbamyl phosphate binds before aspartate does. So that means first the enzyme it binds carbamyl phosphate. It binds the carbamyl phosphate, the first substrate. It binds this. The binding of carbamyl phosphate to the active site of ATCase enzyme will bring a conformational change, which induce a conformational change in the enzyme, which results in a higher affinity for aspartate. So that means the binding of carbamyl phosphate to the active site is inducing the binding of aspartate. So S1, the substrate One will facilitating the binding of S2. So in that way, the binding is cooperative binding. Carbonyl phosphate increasing the binding of aspartate to the active site. So therefore, the carbonyl aspartate, carbonyl phosphate and aspartate both binds to the active site, and the reaction takes place and we will get N carbonyl aspartate, the product. Let us have a look at the structure of ATCase. So like all allosteric enzymes, this allosteric enzyme, that is ATCase, is having regulatory subunits and as well as catalytic subunits. So complete altogether we have 12 subunits in the enzyme. 6 subunits are catalytic subunits and 6 are regulatory subunits. So therefore we have C6 and R6. These yellow circles are the catalytic subunits, and we will have 2 trimers of catalytic subunits. So these 3 are attached to, bound to each other and forms a trimer. Another 3 catalytic subunits will form another trimer. So there are 2 trimers of catalytic subunits, and we have 1, 2 and 3 dimers, 3 dimers of regulatory subunits. So the regulatory subunits are dimers and catalytic subunits are trimers. So these catalytic subunits binds two substrates, substrate 1 and 2. Those are carbonyl phosphate and aspartate. And the regulatory subunits, they bind allosteric modulators. In this case, CTP is the end product. So therefore, CTP acts as inhibitor and ATP acts as activator, because the purine and pyrimidine biosins in a both must be regulated with reference to each other, because the cell always uses purines and pyrimidines in the equal concentration, because in the formation of DNA, suppose if we take the example of DNA, a purine always interacts with the pyrimidine. So there must be a balance between the concentration of purines and pyrimidines. So if the purine concentration is more, then pyrimidines must be synthesized. If the purine concentration is less, then the pyrimidine synthesis must be stopped. So the relative concentration of purine will regulate the synthesis of pyrimidine here. So therefore, the ATP activates. If ATP is more, that means the purine is more. So therefore, the ATP induces the activity of ATCase, and the pyrimidines will be synthesized. Hence, the ATP acts as allosteric activator, and CTP, the end product of pyrimidine biosynthesis, acts as inhibitor. So here we can see that the inhibitor CTP and activator ATP both bind to the same site on the regulatory subunits. So binding site is same. If ATP binds, then it activates, it transforms the enzyme from T to R form. If it binds CTP, so that means ATP is less, CTP is more. So then CTP binds to the allosteric site and it convert R to T form and inactivate the enzyme. So this is very simple mechanism for all allosteric enzymes. Only difference is the allosteric effectors. So here you can see that the substrates carbamyl phosphate plus aspartate S1 and S2. These two are the substrates, and ATCase is the enzyme which forms carbamyl aspartate. So this is the product of ATCase, and so the after the formation of this product, it undergoes several reactions of the pyrimidine metabolic pathway, and finally we will get the end product, that is CTP, cytidine triphosphate. This is the end product. Now by a feedback regulatory mechanism, this CTP, if the concentration of CTP exceeds or goes beyond the necessary levels, then the excess amount of CTP comes and binds to ATCase to the allosteric site, which is located on regulatory subunits. So once the allosteric site on the R subunits bound by inhibitor, allosteric inhibitor, then it induces a conformational change in the R subunit, which also will be transmitted to the catalytic site, catalytic subunit. Therefore, the catalytic activity of the enzyme will be stopped because of changes in the conformation of active site. So once we have excess amount of ATP and less amount of CTP, so that means ATP is more, CTP is less. So that means the purine concentration is more, pyrimidine is less. So in that case, the ATP binds to the same site. It replaces the CTP and binds to the same regulatory site, and it convert the catalytic subunit to a more relaxed form which can bind substrates, and the catalytic activity takes place and enriches the pool of CTP. So this is the mechanism of regulation by allosteric modulators in ATCase. Here, when we look at the curve, substrate saturation curve of allosteric enzymes like ATCase, we clearly observe that if we don't use any product, any allosteric modulator, simply increase the substrate concentration, then we will get this kind of sigmoidal curve. And if we add excess amount of ATP, so then the activity will be increased and it reaches Vmax. If we add CTP instead of ATP, so then what happens? It acts as inhibitor and the activity will be decreased. So therefore, the ATP is acting as activator and CTP is acting as a inhibitor. So this is about the allosteric regulation. Allosteric enzymes are regulated by feedback mechanism, and they are oligomeric proteins. They will have catalytic subunits and as well as regulatory subunits. Regulatory subunits binds allosteric activators and allosteric inhibitors also. They always will be exist. They always will exist in two forms, T form and R form. T form is inactive form, R form is active form. Inhibitor binds to the regulator site and it converts the R form to T form, and the activator binds to the T form and make it to R form, and so therefore the T form and R form, both forms will be at equilibrium. If the activator concentration is more, then the concentration of R form will be more. If the inhibitor concentration is more, then the T form will be more. So it will help in regulation of metabolic processes. So this is about the allosteric regulation. So we have covered all the three different types of regulation mechanisms: reversible inhibition and irreversible inhibition and allosteric regulation also. Thank you.